Welcome to the deep dive. Have you ever stopped to think how a single cell handles all the different messages flying at it? Like 1 minute it's adrenaline, the next maybe a steroid hormone creeping in.
It's pretty amazing. And that's what we're tackling today, the world of cellular receptors. They're not just sitting there, they're active gatekeepers. Information processors, really. They dictate how fast, how strong, how long a cell responds.
Right. And we've gathered some really high-yield stuff here, focusing on the mechanics, the actual speed of these things, and
Yeah.
how they link to real clinical situations, diseases, drugs.
Exactly. Our mission, if you will, is to map out this incredibly complex signaling network for you. We'll hit the major classes, their messengers, and maybe some surprising connections you haven't thought about.
So here's the big question driving this. How can one signal paralyze a muscle in, like, milliseconds, while another signal takes days, literally days, to tweak your metabolism?
And the answer lies squarely in the type of receptor involved. It's all about the structure and what steps are, well, skipped or included in that signal chain.
Okay, let's unpack that. And I think the best way is to organize it by speed.
Absolutely. If we want to make sense of this network, speed is the key organizing principle. It gives us our main categories.
And it gives clinicians a framework, too, right? If you need a drug to work now, you're banking on a mechanism that cuts out the middle steps.
Precisely. So we start at the cell membrane for the faster stuff, and then we kind of move inwards for the slower mechanisms. Okay, top speed, we're talking milliseconds, are the ion channel linked receptors, also called inotropic. They're the fastest because, well, the receptor is the channel. It opens the gate directly, ligand binds, gate opens, boom.
Instantaneous. Okay, what's next down the speed ladder?
Next, in the intermediate zone, maybe seconds to minutes, that's the huge world of G-protein-coupled receptors, or GPCRs. The time lag comes from needing that internal relay race. They have to activate a G-protein, which then kicks off second messengers.
Okay, so there's an extra step or a few extra steps.
Yes. Then we slow down further to the slow category, taking minutes to hours. These are your enzyme-linked receptors. Think receptor tyrosine kinases, cytokine receptors. They take longer because their goal is often changing proteins, usually through phosphorylation cascades, not just simple ion flow.
Right, more complex changes happening inside.
And finally, the slowest of the slow, taking hours to days, are the intracellular or nuclear receptors.
Yeah.
These are playing the long game. They have to get inside the cell, find the nucleus, and actually change which genes are being read. They're altering the cell's fundamental protein production.
Wow, okay. So a really clear sequence based on time, ion channel, GPCR, enzyme linked, and then intracellular. Keeping those four in order, that seems crucial. I actually have a mnemonic for that sequence. It helps me keep it straight. I get everything in sequence immediately.
No, nice. Let's hear it.
So the I is for ion channels immediate, G is for GPCRs, E for enzyme linked, and the final I is for intracellular.
I get everything in sequence immediately? Perfect. Knowing that order really does help predict how quickly a drug targeting one of these might work.
Okay, let's zero in on those fastest ones then. The ionotropic receptors. Milliseconds. What are they skipping to be that fast?
They skip, well, pretty much everything internal. No second messengers, no complex cascades. The receptor itself is the effector. Ligand binds, the protein changes shape, ions flow. That's it. Direct electrical signaling.
Simple and direct.
The classic example everyone learns is the nicotinic acetylcholine receptor, the NATCHR. You find it at the neuromuscular junction, connecting nerves to muscles. It's a ligand-gated cation channel. Acetylcholine hits it, the gate opens, sodium rushes in, potassium trickles out, bang, depolarization, muscle contracts.
And that speed is critical in medicine, right? Like in surgery.
Exactly. You need to induce paralysis quickly for surgery. Muscle relaxants like competitive blockers, think rocuronium, they work by physically blocking acetylcholine from binding to that specific NM-nicotinic receptor at the junction.
So the effect is almost instant flaccid paralysis.
Right. And what's equally important is that you can reverse it quickly, too. You give it an acetylcholinesterase inhibitor, maybe neostigmine. That floods the junction with acetylcholine, which then outcompetes the blocker. Because there's no slow internal cascade, the recovery is also rapid.
It highlights the directness of the mechanism.
Totally. And don't forget the inhibitory side. While nicotinic receptors excite, the big inhibitory channels in the brain, like GABA and glycine receptors, are just as fast. But they let chloride ions in.
Making the inside more negative, hyperpolarizing it.
Exactly, which makes it harder for the neuron to fire. That's where drugs like benzodiazepines come in. They enhance the effect of GABA on the GABA-A receptor, boost that chloride influx, and you get immediate sedation, Anxiety relief, fast excitation, fast inhibition, that's all direct ion flow.
Okay, stepping down one level in speed now. We're in the seconds to minutes range with G protein-coupled receptors or GPCRs. You mentioned a time delay here gives the cell power.
Yes, the power of amplification. See, the receptor itself, that classic 7-transmembrane structure, it doesn't do the final job. It activates an intermediary inside the cell, the G protein. This G protein then goes off and activates another molecule, an effector enzyme, which starts the real downstream cascade.
So why the extra step? Why not just use ion channels for everything if speed is good?
Because this allows the cell to respond hugely to even a tiny amount of signal. Imagine just a few hormone molecules arriving. One activated GPCR can activate multiple G proteins. Each G protein can activate an enzyme, and that enzyme can churn out thousands of second messenger molecules.
Ah, okay. So a tiny whisper outside becomes a roar inside the cell.
Precisely. Signal amplification.
Let's break down the main ways this happens. You mentioned second messengers. What are the key pathways?
Okay, the big one is the CAMP pathway. This involves two main types of G proteins, Gs and G. GS stimulates an enzyme called adenyl cyclase, or AC. AC then cranks out the second messenger, CAMP. CAMP activates protein kinase A, pKa. And G does the opposite. It inhibits adenyl cyclase. So less CAMP, less pKa activity. This push-pull system controls tons of stuff: metabolism, heart rate, think beta blockers acting on G's coupled receptors.
Got it. CMP, what's the other major one?
The other major one is the IP3DD pathway, and this one uses a GQ protein. GQ activates a different enzyme, phospholipase C, or PLC. PLC takes a lipid in the membrane and splits it into two second messengers.
Two from one.
Yeah, and they do different things. One is IP3, inositol triphosphate. It's soluble, floats off into the cytoplasm, and tells the endoplasmic reticulum to release stored calcium. Calcium floods the cell, another powerful signal.
Okay, calcium release. And the other part?
The other part is DLG, diacylglycerol. It actually stays stuck in the cell membrane, and there it activates another enzyme called protein kinase C, or PKC. So you get this dual signal, calcium release and PKC activation, leading to complex responses like smooth muscle contraction or gland secretion.
That's really intricate.
It allows for fine-tuning. And we see this beautifully with acetylcholine again. We said nicotinic receptors are fast ion channels, but the muscarinic acetylcholine receptors, all 5 subtypes, are GPCRs.
Ah, so inherently slower.
Much slower. M1, M3, M5 use that GQ pathway IP3DAG. M2 and M4 use the GE pathway inhibiting CAMPP. This difference explains why activating muscarinic receptors gives you slower, more systemic effects like changes in heart rate, digestion, gland secretion, compared to the instant muscle twitch from nicotinic activation.
And this dual receptor system for HA, that explains the danger of things like nerve agents, right? Organophosphates.
Exactly. Organophosphates irreversibly block acetylcholine esterase, the enzyme that breaks down acetylcholine. So HE builds up massively, and it overstimulates both types of receptors. You get the fast, severe muscle paralysis from nicotinic overstimulation.
Right, the neuromuscular junction.
And you get the slower, but equally dangerous, systemic chaos from muscarinic overstimulation, the SLUDGE symptoms: salivation, lacrimation, urination, defecation, GI upset, emesis, plus bradycardia, bronchospasm. It's hitting both the fast and the intermediate systems hard.
All right, moving on. We're now in the minutes to hours zone, slower responses. What kind of jobs need this time scale?
Think longer-term projects for the cell. Growth, deciding whether to divide, making significant changes to metabolism. The mechanisms here usually involve changing proteins via phosphorylation, which takes a bit more time to orchestrate.
And these are the enzyme-linked receptors. You mentioned 2 main types.
Yes. First, the receptor tyrosine kinases, or RTKs. The insulin receptor is a classic example. Also receptors for many growth factors like EGF. What's key here is they have intrinsic enzyme activity. The receptor is the enzyme, or part of it.
Intrinsic. Okay, so how does that work?
When the ligand, say insulin, binds, 2 receptor molecules come together, they dimerize. This pairing triggers autophosphorylation. They literally phosphorylate each other on specific tyrosine amino acids inside the cell.
They activate themselves by sticking together.
Pretty much. And those phosphorylated tyrosines become docking sites for other signaling proteins, kicking off downstream pathways like MAPK or PI3K. These cascades ultimately influence gene expression and metabolism, but the effects unfold over minutes to hours.
Okay, so that's RTKs with intrinsic activity. What's the other type?
The other main type is the cytokine receptor family. These bind cytokines, naturally things like growth hormone, prolactin, erythropoietin, EPO, many interleukins involved in immunity. Now, these receptors don't have intrinsic kinase activity.
Ah, so they need to borrow an enzyme.
Exactly. They have associated kinase activity.
Yeah.
When the cytokine binds, the receptor changes shape and recruits cytoplasmic kinases called Janus kinases, or JCAs.
Your case.
The JCAs then get activated and phosphorylate the receptor itself, and also proteins called STATs, signal transducers and activators of transcription. These phosphorylated STATs then pair up, travel to the nucleus, and directly bind DNA to change gene transcription.
So again, ultimately changing protein production, hence the minutes to hours time frame, makes sense for things like making more red blood cells with EPO.
Precisely. And while we're talking enzyme links, there's one more specialized system worth mentioning: guanylyl cyclase receptors, like the one for ANP and BNP, the natriuretic peptides.
Right, involved in blood pressure and volume control,
Yeah.
released by the heart when it's stretched.
Exactly. Now, this receptor, NPRA, is interesting because it's not a GPCR. Like RTKs, it has intrinsic enzyme activity. Binding of AMP or BMP directly activates its built-in guanylyl cyclase domain on the inside.
So it makes...
It makes the second messenger CGMP, cyclic GMP. CGMP then activates protein kinase G, PKG. The downstream affects vasodilation, getting the kidneys to excrete sodium and water kick in over minutes. It's faster than nuclear receptors, but slower than the immediate ion channels. It's the heart's way of quickly pushing back against volume overload.
Okay, finally we arrive at the far end of the spectrum. Hours to days, maximum slowness. We have to go inside the cell now, right? To the intracellular or nuclear receptors.
That's right. And the absolute requirement for any signal targeting these receptors is that it has to be lipid-soluble. It needs to slip right through the cell membrane unassisted.
Because the receptor isn't on the surface waiting.
Nope, it's waiting inside, either floating in the cytoplasm or sometimes it's already parked in the nucleus. We're talking about the big players in long-term regulation. Steroid hormones like cortisol, aldosterone, estrogen, testosterone, also thyroid hormone, vitamin D, retinoic acid.
The body's master architects, in a way.
That's a great way to put it. Once the hormone diffuses in and binds its receptor, the whole hormone receptor complex typically moves into the nucleus, if it wasn't already there, and binds directly to specific DNA sequences called hormone response elements, or HREs.
And the point of binding DNA is?
Change gene transcription to turn specific genes on or off, controlling the synthesis of new proteins.
And that's why it's so slow. You're not just flipping a switch or starting a cascade. You have to wait for the whole process. DNA transcription to mRNA, mRNA translation to protein, the protein folding and becoming functional.
Exactly. In a fundamental rewrite, or at least a significant edit, of the cell's capabilities. It takes time to build new machinery. Think about giving someone corticosteroids for severe inflammation.
Yeah, the effects aren't instant. They build over days.
Right. You're initiating a slow, profound change in the expression of anti-inflammatory proteins and suppression of pro-inflammatory ones. It's the long game. Hashtag tag outro.
So looking back, this framework of speed really clarifies things, doesn't it? We've gone from milliseconds for ion channels managing nerves and muscles,
Through the seconds to minutes amplification of GPCRs controlling things like heart rate via second messengers.
to the minutes to hours protein modifications by enzyme-linked receptors like RTKs and cytokine receptors handling growth and immunity.
All the way to the hours to days architectural shifts driven by those nuclear receptors, changing gene expression for steroids and thyroid hormone.
It really covers the whole temporal range of biology. And maybe a final point on location. Some of these systems are more central, some are peripheral.
Yeah, that's a good point. Systems using GABA, or dopamine receptors, for example, are heavily concentrated in the central nervous system, high yield for brain pharmacology.
Whereas others straddle both.
Right. Acetylcholine and glutamate have crucial receptors both in the CNS and out in the periphery, like we saw with AC at the neuromuscular junction. And even dopamine, mainly CNS, has that important D1 receptor in the kidney, causing vasodilation peripherally. Location matters too.
Okay, so we've really emphasized that time equals mechanism. It dictates how cells respond. For everyone listening, think about a common drug you know. Maybe an asthma inhaler, a bronchodilator.
That's likely acting on a beta-2 receptor,
Yeah.
a GPCR. Rely on that intermediate speed, that KMP second messenger system, for relatively quick relief.
Now contrast that speed with, say, the slow multi-day impact of taking a course of prednisone, a steroid acting via nuclear receptors.
It's a completely different timescale because it's a completely different mechanism.
So the provocative thought to leave you with. What other common medications do you encounter? Maybe an antihistamine for allergies. Think about its target, likely a GPCR, and how its relatively fast action relies on blocking that intermediate speed signaling. keep mapping drugs and responses back to these receptor speeds and mechanisms, it really does make pharmacology click.